All right. Well, welcome to this most recent episode of the Akkad and Coca Report. Today, we have the pleasure of having Mr. Dinesh Thakur on. Mr. Thakur is a public health activist who's focused on improving the quality of affordable medicines across the globe. He's most famous now, I would say, for serving for being a whistleblower. He has held some senior positions at Bristol Myers Squibb. He was at the Ranbaxy Laboratories, which is a generic Indian drug company, and he's come to fame for his many years of work, working as a whistleblower to kind of uncover what was happening behind the scenes in the generic drug market, at least in India. Um, uh, I understand, Mr. Thakur, you received a bachelor's degree in technology from Osmania University, master's of science in chemical engineering from University of New Hampshire,
Right.
then graduate training from Syracuse University. So perhaps we can take it from there. You graduated from Syracuse University and ultimately ended up working for Bristol Myers Squibb. Is that correct, in New Jersey? That's where the story starts. Would that be correct?
Well, yeah. So, but first of all, thank you for
Yeah,
inviting me. I
of
mean,
course.
it's a pleasure talking to you both. Um, so, yeah, I mean, I think I graduated from the University of New Hampshire with a master's in chemical engineering, and I had my father's first job was with a small biotech company in Boston called the Genetics Institute, from where BMS hired me. And then I was with BMS for almost 11 years before I got this opportunity to go to India and work at an Indian generic drug manufacturer. So
And what
that
were you
is a,
doing? What were you doing at
yeah.
Bristol Myers Squibb?
So I started working, in essentially when drugs are made, you typically when drugs are formulated, right? Um, you characterize those drugs, essentially sort of analytically test them to figure out whether the formulation is working as intended or not. Um, if you're trying to, for example, make a tablet or a capsule or an injection, how do you know that the formulation is actually working the way it's supposed to work? So there is a department that does analytical testing of that. These are the guys who essentially sort of figure out if you make a capsule, if you make a tablet with the active ingredient and the excipients and the fillers that's essentially going to make it a tablet, do they work the way it's supposed to work? Do they release the drug in the body when it's ingested in the way it's intended to, right? So I was hired in that department as in the Department of Analytical Chemistry, which was responsible for testing and figuring out what exactly was happening to the formulation that was being developed. So in those days, rather than working on small molecules, we were working biomolecules. And two of the drugs that I worked in BMS in those days are on market today. So we were working with monoclonal antibodies, and we were working with essentially sort of biologics at that point in time, figuring out how do you sort of deliver. This is back in 90s and early 2000s. So started there, and then essentially sort of got moved into drug discovery. Um, and then, within the discovery area, again, working in high throughput screening to try and figure out, when you go through libraries of compounds that are tested against a specific target, a lot of this required automation. And my background was in engineering and essentially sort of, PLC controllers. So, we used to build automated systems to essentially sort of increase the throughput and get these things tested properly. So that was what I was doing at that point.
I see. And so, and how many years did you spend at Bristol Myers Squibb doing this?
11 years. So I started up in Syracuse, New York, and then they moved me down to their headquarters down in Princeton, and then I worked at Hopewell, which is a fairly close facility to the R&D headquarters in Princeton, New Jersey.
And so you were kind of ascending the ladder in, is that fair to say, at Bristol Myers?
Well, I don't know whether it's a ladder or not, but the company was very kind to me, so it made me to go to school again when I was at Syracuse, and also allowed me to continue getting a second master's. So that I didn't finish it because they moved me down to Princeton. I was halfway through my graduate program there. But yeah, I mean, so I guess, I was kind of sort of, trying to learn as much as I possibly could.
And again, so your major focus and what you became an expert in, at the 11
Right.
years you were there was how to the process by which you basically take a drug and figure out whether or not it's going to do what it will if you turn it into a capsule, a tablet.
Right.
Essentially, how that active ingredient will be packaged, and
Correct.
that involves, I guess, a lot of animal what type of testing does that involve? How do you do that?
Yeah, so I think that the animal testing is typically preclinical. So this is the second part of my career.
Okay.
When I went into drug discovery and when we were actually sort of figuring out what were the lead compounds, at that point in time, you had to go through preclinical testing, animal models, essentially sort of figuring out, was a drug toxic at this point in time? Typically, animal studies are done to establish toxicity, right? Before you go first in human and try and figure out what is the right dose of that drug in order to address the target that you're trying to work against, right? So this was the second part of my career at BMS. The initial part was more to do with figuring out, once you formulate a drug, whether it's a small molecule or a biomolecule, how do you ensure that the formulation works as it's intended when it's administered in the clinical trial or in the market once it hits the market?
I see. So you're actually getting samples from humans, meaning you're giving different formulations to patients of the drug, and then checking serum levels of the drug to see, okay, is it at the level that we want it to be at?
Exactly. So bioanalytical work is typically that. So when you're testing a drug in the clinic, you obviously would get serum samples, blood samples, and you figure out, what the metabolite, does a drug gets metabolized properly, and you check the serum levels and figure out, what the quantity of the drug is in that point in time. So that's what typical bioanalytical lab does. Yeah.
All right. So great. So now take us to when you made the, I don't know if fateful is the right word, but when you made the decision to leave the peace and quiet of New Jersey to head to India. What happened?
Yeah, so there was somebody of Indian origin who was a senior VP at BMS, and he was sort of on his way to India to sort of assume this role of head of R&D at a Indian generic drug manufacturer. I had not heard of them until that time, but he kind of talked to me about it. He said, "Look, here's an opportunity for you to sort of grow beyond what you did here at BMS. It's a larger role. You can do so much more than what you're doing here, and in a way, you're also going to be able to sort of give back to the country where you came from, to try and sort of help the industry grow there." So that was essentially the incentive, for me to leave and essentially go to India and try and see if things work, right?
So what was the offer? I mean, what was who offered the job to you? And
so he made the introduction. What happened was Rashmi essentially made the introduction to this company because he had already accepted this position as the president of R&D at that point in time.
And so this senior person, his name is Rashmi
Barbaiya.
Okay.
He was the vice president at BMS of the group that does metabolism and pharmacokinetics, essentially early development work. And he was going to go there and become head of R&D. And so he made the introduction. And then what happened was the CEO of Ranbaxy happened to be in the US at that point in time. So he asked me to come and meet with him in New York. I did go meet with him for about an hour or so. And he said, why don't you come to India, see what we do, see what we're trying to do there. And at that point in time, I think Indian companies were essentially trying to climb the innovative ladder. I mean, up until that point in time, Indian industry was essentially a consequence of the drug patent law that India had enacted, which basically allowed them to make copies of generic medicines from the 1970s onwards. But they saw a larger role for the skills that they had developed, which is essentially where reverse engineering drugs. This is what the Indian industry did at that point in time. They wanted to essentially develop new drugs, and that's the reason why they hired Rashmi. And he offered to he said, next time you're in India visiting your parents, why don't you come and talk to our people, see how you like it. So the following year when I went to India, I did start by.
Sorry, and who is this? Who was the CEO at the time, and what year was
Uh,
this?
this was, I think, in 2002,
Okay.
early 2002, and the CEO of the company at that time was a gentleman named Devinder Brar, Brar. He was the CEO at that time. So he met me in New York, and then he invited me to come. And so next time when I was home visiting my parents, I did decide to come, to go to Delhi and kind of, sort of spend a day there talking to people. And it just seemed that it was very busy and bustling. I mean, when you see early stage companies, right? They're just trying to make so many things at the same point in time. There's a lot of chaotic things that were happening, but it looked like a place that could need help, and I knew that I could do something to help them. So,
And to be clear, your senior was going there to, essentially develop new drugs, because that's what
right,
Ranbaxy, I guess, wanted to get into, was developing new
yeah,
drugs. Was that also what you were going to be doing, or what exactly was
yeah,
the role that they had carved out for you, or what did they want you to do?
right. So essentially, my role, the company, what they offered me was had two parts to it. The first part was the work very similar to what I was doing for BMS, which is essentially when I was working in drug discovery, a lot of what we were doing in the labs was generating data. We were running high throughput screens, with automated systems, screening thousands of compounds every day. So you can imagine that, like, tons and tons of data. This is before big data became like the thing in vogue. I mean, we were generating lots of data at that point in time. We were writing software to sort of handle all of that, figuring out what would the lead look like. What would we do with that information, right? Visualize it, for example, and make sense out of it. So part of what Rashmi's challenge in India was this company never had any systems in place. It kind of grew with a span. So he didn't really have any systems or processes in place. So what his offer to me was, come and institutionalize what you did at BMS, build these systems for them, because without these systems in place, it's very difficult to sustain the kind of innovation that I would like to drive in this particular
Dinesh,
company, right?
did they have a library of compounds? Because my
Yeah.
understanding is that all pharmaceutical companies have a huge library, but was that the case for Ranbaxy as well? They had their own sufficient compounds?
So at that point in time, Ranbaxy wasn't actually doing drug discovery, right? Pharmaceutical companies that do drug discoveries have libraries of compounds, right?
Right.
At that point in time, what this company was doing was essentially reverse engineering what was already on the market.
Right.
In order to do that, you don't necessarily need, to have, very large libraries, because you're not screening for a target at that point in time. What you're doing is essentially using chemical synthesis, right, to try and figure out how can I make a simple example, right? So if I want to make a copy of an ibuprofen molecule that is typically used as an over the counter, I don't necessarily need to screen 1,000 chemical scaffolding structures to figure out what ibuprofen looks like. I already know that. What I
Right.
want to do is figure out how can I make ibuprofen in a cost-effective manner, right? What is the chemical synthesis? What chemicals do I mix in order to make a sufficiently pure ibuprofen molecule, right? So this company had skills to do that.
Right.
What it wanted to do was to set up, another organization that did develop large libraries of chemical structures to screen and potentially develop new compounds, sort of new drugs at that point in time, right?
Okay.
So they didn't have libraries of compounds, but what they were hiring Rashmi for was to build this group that actually did the kind of work that pharma companies, innovative pharma companies did in order, to develop new compounds.
Dinesh, can you tell me what you touched on this before, but maybe it's good for us and the audience to hear.
Right.
Why is it that India raced so far ahead in the generic drug
Yeah.
space? So what was it? You mentioned something about the law in the 1970s and that they passed
Yeah.
in India, and so yeah,
Right.
can you tell us, give us that context?
So, what happened was that, in the 1970s, the government of India essentially basically said that we are not going to follow the international standards for patents, right? So patent actually, when you get a patent, you get two things. You get the final structure of the chemical that you're sort of that you're patenting, but also the process by which you actually make that, right? Indian patent law in 1970, the government changed it to say that you can, if you're able to make the same final molecule through a different way, a different structure, right? You would still be able to sort of sell it in the local market without violating the patent law, international patent law. That changed in 2013. India became a party to the TRIPS agreement, which essentially meant that the international patent law would apply to India as well, and this is how it is at the moment. But for 30 odd years, the legal system allowed companies to reverse engineer things. Like today, if there is a drug under patent in the United States, you will not be able to sort of sell a copy of that, because not just the copy of the drug, but the process by which you make is also patented.
Got it.
Right?
I see.
That protection did not exist there. And that essentially gave rise to this industry that essentially became so good in reverse engineering and copycatting a lot of what was available outside of India. So
this will
that's
touch. I see. Yeah. So this will touch on. I mean, we'll get to this in some detail later. But what was the Indian regulatory mechanism in place that I mean, how do we know that India was so good at doing it? How do we know that they weren't selling sugar pills?
Well, so I think that's still a question today, right? So one of the challenges that I still deal with today is the robustness of the Indian regulatory structure. There's a lot to be desired there. It is not on par with what we normally expect, either in the European Union or the United States, in terms of how our regulatory structure works here. Essentially, what happened at that point in time in India between 1970 and late 1990s is that the law allowed a lot of these companies essentially to basically break the chemical down. So if you had ibuprofen, if you had acetaminophen, you could essentially you could break that down to its constituents and remake it, thereby learning what the structure was, right? That is not typically, under the current patent regime, that's not allowed. You can do that, but you can't make money out of it. In the lab, you can certainly do it, but you can't go out and sell it and make money out of it. Why would you want to do it if you can't really make money out of it, right? But in India, that protection didn't exist. So the industry essentially developed these skills, chemistry skills primarily, to reverse engineer existing structures, existing chemical structures. And the industry sort of grew from strength of essentially understanding how chemical synthesis happens. So if you think about chemistry, right? Michael was talking about libraries of compounds. So chemistry in pharmaceutical industry is essentially divided into two areas. One's called medicinal chemistry, the other is called synthetic chemistry. Medicinal chemistry is essentially where you're trying to figure out what is the scaffolding structure of the chemical molecule. Remember, go back to your organic chemistry days, what the benzene molecule looks like, hexagonal ring with, certain things, sticking out of that, what chemical structures look like. Figuring out what chemical structure works against what biological target is what medicinal chemistry is. That is typically what happens in drug discovery. Synthetic chemistry is, once you know what the structure is, how do you then make that structure in a way that reproducibly you can scale it up, right? Making in lab in gram quantities is very easy. When you're trying to make tons of these to make thousands and thousands of pills, you really have to have a good recipe for that process to do that. That is synthetic chemistry. What India has skills, even today, the reason why a large variety of our drugs are manufactured in India is because for 30 years, Indian chemists learn how to do this. The synthetic chemistry side of it. How do you synthesize a particular chemical structure?
Dinesh, is there, I mean, there may be it may vary from compound to compound, but if you have a small molecule drug,
right,
is it that you can have 100 different ways of synthesizing it? Or is it that at the end of the day, there's really 1 or 2 or 5 efficient ways of doing this?
Right. So that's a very good question, actually. You can synthesize a chemical structure in a number of different ways, right? I mean, if you look at chemical reaction engineering, it essentially deals with how you synthesize drugs. The issue is about yield, right? So when you run a chemical reaction, you typically get a certain amount of output, of that particular structure, right? And that's what's called a yield of a chemical reaction. And beyond that, let's say if the chemical reaction runs to completion, it gives you a yield of 70%. That basically means that what you produce at that point in time is 70% pure. The other 30% are essentially, your excipients, your fillers, that impurities that you're generating at that point in time. The point here is that you want to optimize that sequence of steps to make your final product so you get the highest yield, right? You don't want, the sequence of steps, because the chemical, what you produce is not made in one step. It is made in multiple steps.
Sure.
There are lots of intermediate steps that you have to make. The goal is to try and optimize it so that in the end, you come as close to 90% or 92%. That's as good as you can get, right? You don't want to sequence the step in a way that you make 50% of yield, which basically means other 50% is waste, right? You don't want to do that. So to answer your question, there is an optimal path for chemical, for the chemical reaction engineering to actually progress so you get a higher yield. And this is the reason why when we talk about API, the active pharmaceutical ingredient, right? There's a lot of discussion these days about, we're seeing NDMA, what's the rocket fuel, where you talk about angiotensin, ARBs at this point and valsartan, right? Why was this impurity not detected? This impurity was not detected so far because it was below the limit of quantification, right? Limit of detection in this API. So the point here is that these things do happen when you run a chemical reaction. The goal is to try and maximize your yield. So to answer your question, the entire process of synthesis is to figure out what is the most optimal sequence of steps that you take in order to get
right. Thank you. But if I can ask the question slightly differently, suppose you have two teams that are each one is trying to make, a high yield of the same compounds, but they're completely separate. At the end of the day, are they likely to essentially follow the same steps and more or less come up with the same?
Right. So that's an excellent question that you've asked. And let me answer the question this way, right? So what had happened about 10 years ago, most of US-based drug companies actually outsourced synthetic chemistry to both China and India, right? So today, all the large pharma, essentially sort of the chemical synthesis that used to actually happen here at CROs, a lot of the synthesis is done in India and China. What had happened was, what's interesting is that what they did in order to figure out how efficiently these scaffoldings actually work, companies like Bristol Myers, Wyeth, before it became Pfizer, they established relationships with companies in India and China, and they gave them the same recipe and said, we want this compound, synthesize it. Right. What they found interesting was that although the route that which these Chinese companies took and Indian companies took were different, in the end, they converged on the same path.
Okay. Very, very interesting.
So you went back to, so we'll return to our story, our timeline. You went back to India, apparently Gurgaon, right? Gurgaon, which is kind of a suburb
from Delhi.
Miles. Right. A suburb of Delhi. And this was in 2003.
June of 2003, yeah.
And so tell us, you moved your family back and you started and things were chaotic, and tell us how you must have been very excited that you had this new role, and then how did things progress once you got back there?
So we were talking about what my role was. So half of my role was essentially to institute systems to manage information. The other half of my role was I was a head of portfolio management, which basically meant that somebody needed to actually figure out what this company was selling, in what all markets it was selling, right? And figure out what resources to put behind each of these markets. So that was other part of my role. So I had two hats at the point in time. About a year and a half into, about a year into my tenure there, my Rashmi had left. He decided that he wanted to move on, and he had left at that point. But I had a new boss who joined from GlaxoSmithKline. He, a clinical psychiatrist, head of clinical development, gentleman who was responsible for development of paroxetine, Paxil. He came to run the R&D business, and he called me into his office one day and basically said that, look, we need to get a handle on what I mean, what is the kind of information do we have within our files that has been supplied to gain regulatory marketing approvals across the globe? The reason for this was because he was sent to South Africa by the CEO of the company at the point in time to defend an audit that was done by a French inspector, Olivier Leblay, of a clinical research organization this company Ranbaxy was working with, and they found that there was a lot of patient data was fraudulently made up. And so because the drugs essentially were being sold in Africa, which was a WHO qualified country, Raj was my boss at the point. He had to go to South Africa, Johannesburg, and talk to the regulators there saying that, look, we will put this thing right. But on the way back, he essentially was told that, this problem is not unique to Africa. I mean, the drugs that we sell in Africa, but it's larger and much more pervasive. So when he got back, he essentially asked me, I really need to understand what's really happening in this company. Do we really have the basis for which we go and make our marketing authorization applications across the globe? Does the data exist, or how good is the data? So that's where the entire thing starts.
So that give us this important piece of context, I guess. What you described that since the 1970s, India, the Indian government allowed basically reverse engineering. India gained this expertise, but then in order for an Indian company to then sell in other markets, how easy was that? I mean, it sounds like the 70s, the government of India did all this to kind of allow to give access to the Indian population, access to cheap drugs. That's what most likely was the intent.
Yeah.
But so, at what point did Indian companies start selling beyond its borders, and how did the other countries allow that to happen? Meaning, what was the regulatory mechanisms for these other countries?
So, the book actually does a reasonably good job of sort of getting into this area. There are a couple of different things happening in the world at the point in time, right? So one of the things that was happening was the US Congress had just passed what was called the Hatchman Act, which essentially allowed, copies, the generic drug companies to sort of file what's called an abbreviated NDA. It's a shortened version of the new drug application with limited amount of testing in it. So that was what was sort of, the path to getting generic drugs in the US market was becoming much simpler and a lot cheaper at that point in time. The second thing that happened there was, we were going through the AIDS epidemic at that point in time.
So I'll stop just real briefly just to summarize. So in 1983, Orrin Hatch and Henry Waxman, they introduced this bill to allow or to increase the amount of generic drugs that were accessible to at least the U.S. population, population at the time, and at the time, because the only way to get into the U.S. market for a drug was to file what's called an NDA, right? A new drug application. A new drug application required you to run a trial with thousands of patients, maybe, that showed efficacy, not just safety, but efficacy, right?
Correct.
And so that was a big wet blanket, if you will, on generics coming in, because generics then the entry barriers were very high. Not only did you have to reverse engineer and create a drug that was equivalent, you then had to run a trial that showed that this trial was just as
really worked, right? Yeah.
And so therefore they introduced this bill, and this bill introduced a new term instead of NDA, it was called the ANDA, right? ANDA.
Yeah, abbreviated NDA, right?
The abbreviated NDA, which specifically said, hey, you don't have to show efficacy with this new drug. You just have to show
equivalence.
This is it's bioequivalent, right? Right. And so that was one thing. And then the second thing that you're going to describe is also super important: the PEPFAR program in Africa, right?
So,
one more thing on this ANDA. Did they have to, the requirements, were they that the manufacturer of the generic drug would have to show some data in humans that the drug is equally bioavailable and that sort of thing?
Right.
Is that okay? Right. So the sort of thing you were doing for Bristol Squibbs while you were...
Yeah, so if you look at drug development, right, I mean, typically what happens is that once you have a lead compound, and you go into animals and you test for toxicity, and you make sure that dose, it's not, that you really can manage the toxicity of this drug. The first thing you try and figure out is what is the right dose, which is usually the phase 1 study, right?
Mm-hmm.
So you essentially figure out, in human, healthy human volunteers, healthy human patients, what is the right dose for this drug? Where do you see a dose response when you give this drug for the indication that you're studying, right? That's essentially what was required of the generic companies. Basically, what the regulation said was, look, as long as you can prove that the dose response for your formulation, meaning the formulation that the generic drug company makes, is similar to what is already, the innovator company has already done that, right? I mean, this is something that they have already done it. This drug has been on the market for X number of years. We know exactly how this drug behaves in the human body, right? Now, a generic company, if you can formulate your drug, your version of the drug, and as long as it behaves in a manner similar to the innovator company, we will give you an approval to market it. Now, what the US FDA did was it essentially gave a kind of a range. It said that okay, if after a period of time, and your physicians, you know how drugs get metabolized when you give it to the patient, the level of concentration of the drug in the plasma increases, right? And then after a while, your bodily functions start to excrete it out, right? Absorption takes it out, kidneys essentially filter things out, right? And the drug is excreted out of the body. And so you will essentially see a sigmoidal curve, just sort of that concentration increases, and then after a while it starts tapering off.
Right.
The US FDA said that, for the generic company, as long as your drug, your formulation mimics that, plus or minus 20%. So going from 80% to 120% of the concentration of that drug in the human body, as long as you're able to do that, we know that this drug works. Tell us how you'll make it, right? You still have to, do what's called the CMC, the Chemistry Manufacturing Control Section, which is essentially the manufacturing area. But you don't really have to run a large-scale study, clinical study, right? You don't have to demonstrate safety and efficacy because that's been done before. All you need to do is demonstrate equivalence at that point in time. That is what the regulation said.
Very good. Thank you.
Right? And so with these, when these regulations came into the picture, generic companies, sort of knew that this was a path that was available for them to enter the market right now. I mean, essentially the threshold for entering the market was brought down significantly compared to what it existed before. The other thing that actually happened was when, George Bush became the president, we were going through an AIDS epidemic, and the antiretroviral drugs at that point in time that we used to control, the platelet counts and ACD counts at that point in time were very expensive. So one of the things that President Clinton did was he tried and brokered this deal, which basically said that the companies based overseas in India, because of the cost of labor and the cost of materials and things of that nature, if they can make these medicines at a cheaper price, right? And then they were distributed to patients in Africa, where they were people, you were seeing a very large disproportionate effect of this epidemic, then we'd be able to do some good about it, right? So he basically bought, the Indian generic companies and USAID, which was essentially running the PEPFAR program, with US taxpayer funds. Both of these organizations together. But then one of the things that the people who are running PEPFAR said, well, how do we know that this stuff is actually good? I mean, we've never actually bought these kind of medicines that were made overseas, and they really have a very bad reputation in terms of the quality of what these companies produce. So one of the things that they wrote into the law was, well, if you were to apply to the US, to PEPFAR program as a part of the USAID, we needed our FDA to bless you that the quality of what you're producing is good, right? We don't want to spend our money, buying stuff that doesn't work. We want to make sure that these medicines, if you're going to use our taxpayer money to send to Africa, we want to make sure these medicines are of good quality. And so that basically opened up a doorway, a pathway for Indian genetic companies who basically said, well, if we can do this, and we can demonstrate that our antiretroviral drugs are as good as anything else, why can't we do it for other therapeutic areas, right? So that opened this floodgates of ANDAs. And now, if you look at the US FDA annual report, they actually report on how many generic drug approvals have they actually given every year. That's a metric that's measured in terms of productivity of the US FDA, right? So that opened the floodgates to the generic drug market within the United States.
Very good. So, and just to be clear, the PEPFAR program was going to purchase would purchase. I mean, that would be they would buy the drugs from the generic manufacturers, provided
overseas, yeah,
right, and to be sold in Africa predominantly.
To be essentially dispersed in Africa. This was essentially a program run by USAID, to sub-Saharan Africa, African countries. Yeah.
Okay.
So that's the context. So this is where Indian drug companies, by the time you get there, are selling drugs to all these different markets all over the place. Correct?
Well, this is just beginning too. I mean,
beginning to, okay.
Right. So the thing that I mentioned to you, what caused all the furor at WHO was this drug called lamivudine. It was one of the NNRTIs, which was essentially, this was being formulated by Ranbaxy laboratories at that point in time to be purchased with US taxpayer funds as a part of the PEPFAR program to be sent essentially to Africa. And so basically that is where the entire thing got started. And then, although there were generic companies in the US and they were already doing this, but the invasion of Indian generics into the United States market essentially began with, the law that allowed them to essentially lower the threshold for market entry. And, these things, came around the same time, creating an opportunity. Right. And then, you could look at every patent expiry and say, well, can we now make that particular product? Because we know the moment the patent is no longer valid, we can enter the market and we can compete on price and we can bring the price down significantly. And that's essentially how the process is.
So what was the in Africa? What was it that initiated the problem exactly? What's the tampered? Go ahead.
Yeah. So even before the US got involved, right, even before President Clinton actually got, this whole program together, countries in Africa were buying drugs from India, right? I mean, because India had a very large pharmaceutical industry and many of the African countries did not have an indigenous manufacturing capability. They were buying drugs from India. But one of the quality standards for them to purchase drugs from India was what was called the WHO pre-qualifications list. So this was sort of a third party, standard that the World Health Organization had set up to say that if, the World Health Organization came and inspected your facilities, gave you a stamp of approval, at least that could be valid by the purchasing organizations of countries like South Africa, Congo, Ethiopia, and which were essentially buying medicines from India. They looked at the WHO and say, how do we know that these companies that are located in India can produce a quality product? Well, what the WHO did at that point in time was they established a pre-qualifications program. It was led by a gentleman named Dr. Andre Van Zyl. And he, along with Olivier Leblayer, who was a French inspector, they were essentially the hands and ears of the WHO. They would go into these facilities overseas and inspect these facilities, right? And tell them, look, here are the shortcomings that you have. This is how you make a quality product. And once the WHO gave them approval, meaning that that site became pre-qualified for the WHO procurement, then other countries would look at that and say, well, if the WHO says this is a good site, they know what they're doing, they're making good quality product, we would then ask them to bid on the tenders that we would have to purchase, lamivudine, stavudine, all the antiretroviral drugs of it. So that is how the process worked. What the Clinton Foundation did was, because they bought, scale to this, right? They bought much larger distribution, capability. Rather than each individual country in Africa buying for their own purpose, they said, well, with USAID, we will leverage, we will buy in scale, right? Therefore, we will bring down prices. This is what President Clinton did through Clinton Global Initiative. He bought the Indian companies which were making these drugs, and the African nations were buying these drugs together and said, we will facilitate this process so that you can now lower the price even further and you'd get good quality medicines, supported by the US taxpayer funds through a PEPFAR program.
I see. And so, there's some irregularities that are found by this French inspector in Johannesburg. I guess these were factories. Anyway,
these were factories in India. They were supplying drugs to Johannesburg.
So that means that he had gone to India,
inspected the facility,
exactly. and he found some irregularities. So he reported this. Your boss went down and got word that, hey, and your boss, by the way, is also a GlaxoSmithKline guy,
right?
And he tells you, hey, Dinesh, you have to give me a report of what's going on here. So how did you start to do this? Yeah. How did you, I mean, that's...
Yeah. So, basically what I had to do then is essentially I had to set aside the other part of my job, because this was essentially what I had to do for the next 4 or 5 months, to trace essentially every piece of data. So if you think about, the kinds of data that is actually generated, right, when you do drug development. So just give you a simple example. So, for example, when you make an application for market authorization in Africa, in South Africa, the South African regulator, MCC, basically says, okay, tell us how would you make this particular drug, right? Tell us what the recipe is. Tell us how would you control, right? So, for example, let's assume that in your home you're cooking a pot of chili. For 2 people, it's easy to make, but if you're making it for a big gathering, if you want to go to a picnic with 15 people, well, you have to have a process in place in order to know how much salt you're going to put, how many tomatoes do you need, right, and all that kind of stuff. So that is the chemistry, manufacturing, and control section of the ANDA, which basically says, okay, look, this is how we're going to make this particular drug, right? And then they want to know, have you tested it? Is it bioequivalent, right? Does the drug work in patients, although a limited number of patients, but you still have to test and show that it is equivalent to an innovator drug. So all of this data essentially gets compiled into what's called a marketing authorization application. And what I found out when, in the exercise that I was asked to conduct, was much of this data didn't really exist.
How did you know that?
So one of the things that actually happens in drug development is that every lab that actually does something, they have to record it into a lab notebook, right? Yeah. And the lab notebook records your hypotheses when you do an experiment. It tells you exactly how the experiment is done. It tells you what the results of those experiments are, right? So the first thing to do is to try and locate those lab notebooks and figure out whether the experiments actually done. If somebody tells you the experiments were done, the second thing you look at is that what was recorded in that lab notebook and what was reported in the dossier that was submitted for market authorization. Do they match up, right? The issues that you run into is that regulations are different, right? For example, the US FDA, even today's regulation, right, says that for general drugs, not narrow therapeutic drugs, right, but general generic drugs, you can see a variation of 15 to 20%. So 85 to 120% variability in bioequivalence is accepted. You go to India and you start looking at Indian regulations. Indian regulations say 30% on each side. So 70 to 130% is acceptable. So there are variations of this nature. So even if you have the data, you have to then figure out, are we complying with the local regulations where we're actually selling this kind of stuff, right? You go to the clinic. I mean, you've written a report that says that we have tested this against 24 patients. 12 of them were given the innovator drug. The other 12 were given blindedly our formulation, the one that we've made. And we've drawn blood samples and we sent those blood serum samples to a bioanalytical lab, and we've looked at exactly how much, of the compound is in there, right? Now, I know the report exists. I know the report says we've got 24 patients. But if I want to verify that, I first go and say, okay, show me the informed consent when you enroll these patients, right? Show me, when the admission forms, right? When were they dosed? What was the dosing time, right? And I need to understand, what was the blood sample, the sample ID that was collected? Who analyzed it? What kind of bioanalytical work did you do on it, right? So you got to trace all this stuff out. Every step has to be traced out. Stability, like for example, you basically say, okay, my formulation, if you go to Caremark or Walgreens today, it tells you this drug expires at this date, right? Do not take after this date. Well, how do you know that formulation expires at that date, right? Have you tested it, right? I mean, so there is this whole notion, whole area called stability, which basically says that once you make this drug, you're supposed to sort of put this into a temperature and in humidity controlled conditions, and you take samples a month, 3 months, 6 months, 12 months, 18 months, and 24 months, and you test it and say how much of the drug is still there, how much of it is degraded. If it is a solid oral dosage. Now, if you think about if it's a liquid formulation, right? Think about this. This stuff settles at the bottom, sitting there at 18 months, right? What kind of instructions do you actually put on the label, to sort of re-homogenize it before you actually dose that? So there's a lot of stuff that goes in, but all of this is recorded. One of the things about drug discovery and development is every piece of information is recorded someplace. So you backtrack and trace all of this data back. And
what happened when you tried to find what happened? What happened? So did the notebooks exist?
Well, no. So in many cases, notebooks didn't exist. In many cases, notebooks were half filled. What we found out that they would actually run. For example, they would put all the samples for stability in the stability chambers, but they would draw all data points for 1 month, 3 months, 6 months, 12 months, 18 months the same day and analyze it, and essentially say everything passed. So things of that nature, right? So basically we had to sort of, do a little bit of detective work to try and figure out. I had thankfully a team of 6 people who I work with, all great guys. Each of them were assigned one area of the world, and they essentially kind of dug deep into that specific area, that specific market. And we found out that, depending upon how tight the regulations were, right, the level of information that the company actually had varied. For example, in India, where we knew that the regulators didn't really care, there was basically no data. In the United States, when we knew that the FDA was a good regulator, they would at least find out stuff, about 50% to 60% of the data still existed, and a lot of it didn't exist. So it was a gradation. It was not like, you can't paint it with the same brush all the countries. So that's what we found out.
Oh, wow. And so were you surprised?
I was. I mean, this was not something that that I would have expected, right? I mean, this is not
right. Tell us a little bit about Ranbaxy. By then was a big company. I mean, right? It was making news, and it wasn't like just an overnight a fly-by-night operation. It had received a lot of press and so forth.
It did. And I think, I mean, the company essentially was on its way to become $1 billion company at that point in time. They were making upwards of a couple of $100 million in the United States, and they had a very strong portfolio, what is called first to file. I mean, so if I could just take a minute to explain how this thing works, one of the things that the law that Dinesh talked about in 1984 did was it incentivized the first company to get an approval for the generic in exclusivity of 6 months.
That's for the U.S. market, right?
For the U.S. market, and that's a big incentive. So when Lipitor went off patent, when atorvastatin went off patent, the first generic, which Ranbaxy had, made $600 million in the first 6 months. I mean, Lipitor essentially was a $10 billion drug. But, this is very lucrative. And so they had actually a very strong, probably the best pipeline of first to files in the United States at that point in time, which was valued very, very highly. Mm-hmm. And so the company was essentially was a known entity, a known name in the generic.
So, and I guess that's why folks like you and folks like me would be surprised, right? I mean, if there's this much cash that you're generating, and there's that much that's at stake, why wouldn't you want to get these things right?
Well, so I think that's $1 million question, right? So think about it this way, right? If you've grown up in an environment where you were never actually held accountable, remember these companies essentially grew up servicing market in India, right? Where regulations don't really exist, the regulator is incompetent, and you can buy your way out of pretty much anything that you want to do. If that's the way that you've been born and brought up, just because you now work in a different market, you don't just change your colors all that quickly, right? So you sort of try and figure out how do you beat the system. So that was one aspect of it. The other aspect of it was that the company's ambitions were very big. They wanted to be, in every market, selling every, servicing every need at every point in time. But what happens is that, you need to be able to sort of have that capability to develop those kind of formulations. I'll give you a very simple example. One of the things that big pharma companies, innovator companies do is that they don't have the same strength of their product in every market. The reason is very simple, because they want to protect their pipeline, right? So they may sell 20 and 40 milligram atorvastatin in the United States, but in Europe, they may sell 5 and 10 and 15 milligrams of it. So although, somebody may get an approval to make 20 and 40 milligrams, if they don't have the technology and the process to make 10, 15, and 20 milligrams, they won't be able to get into the European market. It's very simple. It's a strategy that the big pharma companies use to essentially evergreen their patents. Now, if you want to be in the United States and also in Europe, then you have to have the capacity within formulation development, within analytical, within process chemistry, to develop all of these formulations, not just the 20 and 40 milligrams from the US, but you also have to develop the 5, 10, 30 and 60 milligram formulation as well. So what this company was doing was it was never actually investing, in actually developing all of that. It was just taking data from, what it was doing for the US and copycatting all of that and basically saying, okay, well, this is the same data that we've got for our other product, strengths that we have in Mexico, in Brazil, in Romania, and things like that. That's what they were doing. So, I mean, it's a combination of several things. It's just, because they could get away with what they were doing in India, and because the market was large enough, they could see how lucrative this thing was, but they just didn't want to sort of make that investment in actually making it happen, doing it the right way. This is basically what led to all of this.
So you came up with a I guess a presentation that meaning,
yeah.
Did you how did it work? Did you tell your boss? Your boss would ask you. You showed him the presentation. Was he surprised?
Well, so before the presentation, what happened was before you actually put stuff on slides, you have to collect all of this data. So we were using spreadsheets, extensive spreadsheets. Essentially, every data point was sort of mapped out in that spreadsheet for development, for formulation development, analytical, process chemistry, validation, all of that kind of stuff, right? So as and when we were collecting data, my team was, I was not the only person doing this. I was the one who was collating data, but my team was the one who actually went to all these labs. And, some of these places were like really, really far off. They were not in the same place. So these guys would travel, talk to people, collect all this data, put it into a spreadsheet, and we would collect all that. And as an intermediate progress report, I actually, went and talked to Raj about it and showed him this and said, look, this is what it looks like. So he basically said, continue working. At one point in time when we had enough data, he asked me to put together a presentation. He said, I'm going to go and talk to the board about this because it's just getting out of hand. So I made this, 24-slide presentation, basically sort of going through what our findings were. And then, Raj basically went to the board and made the presentation. Unfortunately, they didn't really want to hear him.
So Dinesh, at this point, where your thinking was that, did you sort of think that what you just said, that this is primarily a cultural thing in a way, that the whole company, they're not used to doing this, but you thought that the board would be, you would want to know that, or would want to have their eyes opened, that it was just primarily that, the way of doing business was just had never been inculcated at all the different levels of the company.
Right.
And
were
so
you
expected the board to sort of wake up and recognize the importance of that factor.
It was not me so much. I was in the weeds. It was my manager, my boss, Raj.
Okay.
He was the one who essentially was dealing with the board. So as the president of R&D, he was the one who basically went to the board to essentially, in the hopes that when he made the presentation and asked for authority and resources to put it right, that the company and the board would would empower him with the ability to fix this problem. That was the goal that he had in mind.
So what was the reception? So it was just him presenting to the board, and it was your data that was presented.
Yeah, it was a subcommittee. It was not the board. They basically sort of narrowed it down. They wanted to be widely known. So they essentially had assembled what was called the scientific committee, which was one of the subcommittees of the board. But it was adequately represented. I mean, the CEO was there, the head of business was there, members, the chairman of the board was there, a couple of other key members were there. He made the presentation. Basically, they asked him to get rid of it. They basically said destroy the whole thing. And
wow.
Not just the presentation, but the computer on which he made the presentation. Somebody took a hammer to it, and so that was basically it. And Raj, I mean, he's one of my sort of heroes, and basically said, look, I'm a physician. This is just nonsense. I can't be working in an area that puts people's lives at risk. I took an oath to protect and treat people. This is nonsense. So he basically resigned 2 days after that presentation. And so, he left, although that was not widely known, so there was also a lot of other corporate shenanigans going on, because the family wanted to sort of the sons of the founders to become the CEO. So there's a lot of churn up there. Raj left. And so, basically they made my life miserable as well. And then I had to leave after that. And that was the end of Ranbaxy.
Wow. And at some point after that,
yeah,
while you were still working for Ranbaxy, or after you had left Ranbaxy, you then reached out to the FDA. Is that right?
Yeah. So I left Ranbaxy in April of 2005, and basically I thought I was done with it. But unfortunately, I couldn't sort of live with what I knew, because I mean they were still doing the same nonsense that they were doing. So I reached out to the WHO, the US FDA, and a few other regulators who I knew had conclusively known that their markets were essentially being sold chalk powder by this company. Unfortunately, WHO wasn't very interested in pursuing what I was saying. Neither was the UK regulator. But thankfully, there was a gentleman named Edwin Riviera Martinez. He passed away. He was just such a wonderful gentleman. Edwin was the head of compliance at the FDA. He was the one who sort of took it upon himself to investigate and figure out what I was saying did really actually exist, or I was kind of making baseless allegations. So,
Dinesh, I think I don't know that we can know the answer, but I think I know what the answer is. But my question is this: can we tell? I mean, what was not in place? I mean, there was no quality control documentation. But do we have an idea of what was actually being produced by Ranbaxy? I mean, how effective was it? Completely ineffective? Was it 0%, or was it 80%? Or I mean, these drugs were being sold. Presumably, some patients were responding, so it was not completely ineffective.
So I don't know. I don't know the answer to this question, because you see, one of the biggest challenges that we still sort of deal with is we really don't have good surrogates for bad quality drugs. I mean, I'll give you an example. I work very closely with Dr. Harry Lever, who is a cardiologist in the Cleveland Clinic. He essentially is a specialist in hypertrophic cardiomyopathy. And he's one of the clinicians that has systematically documented the effect of substandard drugs. Because you see, when you're dealing with patients that are that sick, it's very difficult to differentiate, issues that you run into because of the natural progression of the disease, right? What is really happening with the patient versus what you're seeing with the drug, and if the drug is working or not working, right? And adverse events it's producing. And in a country like ours at this point in time, with the advanced systems that we have, the knowledge that we have, if we have trouble in the medical fraternity to try and understand and differentiate between these two causes, imagine what public health officials in third world countries do, where they're buying these drugs, which essentially are nothing more than chalk powder in many, many cases.
Hmm. But clearly just to add on to Michelle's point, I mean, the PEPFAR program is a success, correct? I mean, there are millions of people that are living with AIDS, controlling their.
You're
HIV.
right. You're right. So there's clearly no. So I mean, I think that this was some of the discussion on Twitter when the book came out, where people who are involved in the program basically pushed back, basically saying that yes, I mean, if the drugs were so bad, why do we see so much of a remission rate, in patients? And I don't have an answer to that. I mean,
Right.
could it be because so my experience is this, right? I don't think that every batch of the drug that these companies produce is bad. I don't believe that at all, because the companies actually have the skill to be able to make good drugs. And many of them do. But the problem is that when somebody is not looking over their shoulder, they take shortcuts. So for example, I'll give you an example. Harry uses this drug, metoprolol because the patients that he uses, they have, basically, just to try and control, the issues that he's dealing with. One of the issues with this drug is that it has renal side effects. And if you don't, some batches of these drugs made by certain companies essentially cause severe renal side effects where the patients bloat. The kidneys don't process urine at all, and you see significant hospitalization issues with it. Now, does it happen with every batch of the drug? No, it doesn't. But it does happen with reasonable frequency of this particular drug. And we also see this with nanotherapeutic dosage drugs. So levothyroxine, right? So if you actually treating thyroid conditions, for example, anything to do with organ transplant rejection drugs. Nanotherapeutic drugs, dosage drugs, are highly susceptible to these kind of minor changes in quality control processes. So the answer to your question is, does every batch of the drug, do you know if every batch of the drug is bad? No. The issue is, how do you find the batch-to-batch variability?
So since all this has happened, basically to move the story ahead, whistleblower to the FDA. The FDA investigated. The FDA found, basically found what you had said to be true, and ultimately Ranbaxy, had to pay a penalty, the largest penalty to date, perhaps $500 million, correct?
For a generic drug maker, yeah.
For a generic drug maker, sorry, and Ranbaxy essentially is it still functioning? It's not a.
No, so it was bought by a Japanese company, Daiichi Sankyo,
right?
But then it turned around and sold it to another Indian generic drug company called Sun Pharmaceuticals. So it's kind of merged into Sun Pharmaceuticals.
Okay, all right. But and so is the upshot of this, and the upshot of this is, of course, the U.S. FDA has stepped up its regulation and no longer gives warning by sending a delegation.
No, that's not true. That's not true.
Oh, that's not true. Okay, good. No. So tell me what has changed for the better?
No, I think that we've learned a lot, right? I think that there are people like Peter Baker, who was the drug inspector in India and for a long time in China. So this is the back shot of this, right? So if you just give me a minute, we'll try and lay out the issue that we're dealing with. Remember, when the Food and Drug Act was approved here in the United States in the aftermath of the thalidomide tragedy that we had, the preponderance of manufacturing was in the United States, right? So when we wrote our regulations, we basically said, look, we are going to rely on a system. What we're going to say is that we are going to tell the manufacturers, tell us how you make these drugs, right? Under the penalty of perjury, tell us, this is what the process is that we're going to make. What we're going to do as regulators is we're going to come unannounced to your facilities, we'll knock on your door, and we'll verify that you're following your own process, your standard operating procedures, right? That's typically how the FDA works. It's an honor system. This system worked great, until about late 1990s and early 2000s, until everything was in the continental United States. I mean, the site that I worked in, at Bristol Myers Squibb site in Syracuse, New York, was the last site that made penicillin, in the United States. We don't make penicillin anymore, so all of this is gone. I mean, we had manufacturing facilities in Puerto Rico at one point in time. Puerto Rico had the largest number of manufacturing facilities that made pharmaceutical products. All of that is gone right now to India and China. The point is this: the basis of our regulation was an honor system, where the food inspectors, the drug inspectors, essentially come unannounced and verify what you say that you will be doing in your application for market authorization is what you're doing. Unfortunately, when the facility is located overseas, that's not possible, right? I mean, an inspector can't fly into India without a visa. They can't go into China without a visa. Even if they go to India with a visa, they're not going to be fully functional the first couple of days because of jet lag, right? It takes time for them to get back on their feet. They don't necessarily know the local language. They don't know the layout of the place. So you rely on the company to chaperone you. They kind of know exactly where you're going to be, right? So these things, I think, will have to change. I mean, when Altaf Lal was the India head, FDA India head, he had run a small program where he basically essentially put the element of surprise. He took away this notification saying, we will tell you when we come to inspect you, right? And during his time, when Peter was there, they essentially found out the largest number of violations of good manufacturing practices. Unfortunately, that program was shut down. And today, the US FDA still gives an early notice to all these facilities in China before they go inspect.
So Dinesh, you don't think, I mean, it sounds like you don't think that what you discovered is unique, was necessarily unique to Ranbaxy. What are your thoughts on that, actually?
Well, I mean, I think that it's not about me saying whether it's unique to Ranbaxy or not. Fortunately, what the FDA does is it publishes its inspection findings on its dashboard. So if you go to the US FDA website and look for inspection dashboard, the data is a little dated. It's usually up to date until last quarter, but nevertheless, I mean, all that information is available. Now, if you look at that information, you'll see that a very large majority of the warning letters, which is essentially a punitive regulatory action that the FDA has sort of implemented, those numbers are so high when these facilities are overseas, right? Inspection observations, these are so high numbers when the facilities are located in India and China. And so this is not a one-off thing that we saw with Ranbaxy.
Sure. I mean, I'm certainly. It's easy to believe. It's. Can I ask you now to comment a little bit on what we've lived? This is such an opportune conversation for me because in the last, for many in our audience, especially the cardiologists, in the last year or so, we've lived with these recalls of ARBs. And just actually a few months ago, I asked somebody who had published on this. I asked, well, did all these recalls because of impurities of this carcinogen, NDMA, we haven't seen them in the brand name Diovan, right?
Yeah.
Or so. And so is Diovan entirely manufactured here? What keeps Diovan from manufacturing elsewhere? And so can you clarify that? Because I actually have no idea.
Yeah, so I don't quite know where Novartis makes Diovan.
Okay.
Although, so there are some challenges with the manufacturing process, right? So if you look at the regulations for generic drugs, the generic drug regulation says it has to have the same active ingredient. But they can substitute excipients, basically, the filler material that holds the matrix together.
Right.
That doesn't have to be the same exact filler material that innovative drug manufacturer like Novartis uses to make the ARB, right? Now, the issue here is as follows, right? I think that what we're seeing with NDMA, from what my understanding is that this is a product of degradation. Essentially, you're seeing, when the drug sits over a period of time, you're seeing cross-reaction between chemical moieties of that particular molecule.
Mm-hmm.
And it exists in certain below the threshold area level that could not be detected when Diovan was the ARB that was being sold. what we're seeing right now is higher levels of this, in drugs like ranitidine, which is, omeprazole. You're seeing it in, valsartan, olmesartan, all the sartan drugs. The point here is that, the manufacturing process is not identical to what Novartis uses. The question there is, number one, what is changed, right? I mean, in this particular case, it was very obvious because, we were using recovered solvent in manufacture of API, which is essentially what introduced NDMA into valsartan at that point in time. The long-term effect of this is, I do not know, because even today, the FDA says continue taking the drug. The regulatory advisory is not to stop taking any of the sartans.
Right.
So we will have to pay more attention to this. I don't have an answer for you.
Could you actually clarify for me? Because how was it discovered? I mean, is there a process by which batches are periodically tested by the FDA?
No. So the way it was discovered basically was that, so we don't have the ability to actually test batches. We do not test anything that comes into the United States. The way it works is that when a consignment of drug that is manufactured overseas comes into the United States, the only inspection that is done is a DEA license. Majority of the air shipment comes into Dallas. That's the port of entry for us. From there, it essentially goes to our distribution warehouses in Arizona. So Express Scripts, Medco, all the big PBMs, they essentially have large warehouses where essentially repackaged for retail, and it's essentially shipped out from there. We do not test any batch. The thing that has changed is that there is one pharmacy called Valisure. It's a Connecticut-based pharmacy. They have established a business model where they actually test every batch of the drug. And then what they do is the tested batch of the drug is what is essentially dispersed to the patients. That model, I think, has taken off. Valisure and David Light were the ones who actually discovered NDMA in the BRB drugs.
That's really remarkable. Because now it raises all kinds of questions and concerns. It's unbelievable.
It does.
So Valisure, not the FDA, is the ones that discovered this.
That is correct.
Wow.
I mean, they basically brought it to the FDA's attention. Obviously, once they found out,
right,
and they continue to investigate this particular issue, not just with the ARBs, but with ranitidine now, and we're seeing this impurity in other areas as well. The point here is that this is as follows, right? So, I mean, I have seen in the medical literature streptokinase, which is essentially also used in your practice. The active ingredient in streptokinase, all the way from 80% down to 20%, depending upon where you actually buy the drug, whether you buy it in the UK or the US, or you buy it in Pakistan, where it just becomes zero at that point in time. So manufacturing actually has a specific outcome, in the way that we control these things. But we are now seeing emergence of different models. Like Valisure's model is very different. I mean, for $20, I think, for a 30-day prescription, they actually test that drug and send you that tested drug.
Right.
We may have
an episode on Valisure, because we just recently discovered, and we thought it was extremely interesting. So, but just to address that point, it's, as far as you know, as a chemist, it's possible you can take a batch. At least technologically, it's possible to take a batch, analyze for stuff that shouldn't be in there, that is known to be toxic.
Mm-hmm.
But that's different from actually knowing if the batch is going to be really as effective as it claims to be, or because it may have, right? Yeah.
So the way it works is that every batch of drug that is manufactured by the manufacturer, before it's released to the market, goes through what's called release testing.
Okay.
And so this is the manufacturer's responsibility. What they're supposed to do is they're actually supposed to test for potency, which is typically what assay is, impurity profile, which is the filler stuff that you put in, to what extent is that acceptable, right? They also provide stability of that particular formulation. So, and at the end of doing all of this, they are supposed to issue what's called a certificate of analysis, which is a legally binding document. So it basically says that, look, here are the tests that we have done. This is the result that we found. This is the range in which the result's supposed to be, right? And this is in the range, right? This is typically what happens. Now, remember, you see things like recalls, right? How do you actually have a recall? The reason why you have a recall is that when a product is in the market and the product degrades, and one of those results actually goes out of that specification, that triggers the recall. But the thing is, there is a process in place for you to actually measure the quality of the product before it goes out of the market.
Right.
But that is done by the manufacturer.
Okay. And that supposedly was supposed to be in place for the generics in India, for Ranbaxy and all of them are supposed to have. Yeah, we have a tendency, and it's erroneous, but to think of the synthesis process as being one thing, and then quality controls being something separate. But really, manufacturing is one process at the end of the day, where it has to be, not only do you have to manufacture the stuff, but you can't really say anything about it until you've done the quality controls at the same time, right?
So quality control, you can't think of quality management as independent of the manufacturing process. It has to be a part of the entire manufacturing.
So, Dinesh, do you think the solution lies in bringing manufacturing back to the United States? Do you think the solution, as we've talked about, is something like, is third parties like Valshore?
So there are a couple of different things, right? So I'll give you those are two good examples. So Valshore is one example where they're essentially backfilling a gap that we have in our regulation, because our regulation doesn't require us to test batches of product. This is a private business that's beginning to do this, right? Mm-hmm. Whether that takes off or not, I think we'll have to see, but they're providing a service that's sorely needed. The other thing that is also happening is that people are bringing manufacturing back into the country. This is an organization called Civica RX. I don't know if you've looked them up, but Civica just started manufacturing vancomycin back in the US again. It's essentially run by a bunch of people from the industry who have essentially retired, but they've come together now. They are essentially taking over old manufacturing plants and recommissioning them to manufacture vancomycin, in the United States, because that was a drug that's on the shortage list for now.
Michelle, we have the chance of getting retweeted by Donald Trump if we talk about this. manufacturing coming back to the United States. I'm sure he'll take credit.
Yeah,
that's really fascinating.
So there are things that we need to do, but I think that we need to look at this holistically, right? I mean, last week the House Energy and Commerce Committee had a hearing where they focused primarily on the API in China, which is one aspect of it. API in China certainly is one aspect of it, but I think formulation in India is another aspect of it. Manufacturing, the drugs that are on the shortage list, because those companies in India and China don't want to make it anymore because it's not commercially viable for them, right? So somebody will have to bite this bull, take this bull by the horn and do something about it. And there have to be incentives for business to be able to reset up these things. And I mean, it's interesting, right? So if you were to set up a new manufacturing facility today, you wouldn't do it the old-fashioned way, right? You would actually employ a lot more automation. You would use continuous manufacturing, right? You would do it in a very different way, and you probably be a lot more cost-effective compared to the way BMS made penicillin when I was working in Syracuse at that point in time.
Fascinating. Dinesh, can you tell us? Sorry, Michelle, go ahead. Sorry.
No, nothing. I mean, it's just unbelievable. The opportunity, it seems all of a sudden we realize that we've been asleep at the wheel for the last 30, 40, 50 years. And what's fascinating to me is that really a lot of the push for generics and whatnot comes from, legislations and things like that. Because to the lawmakers, it's like, oh, it's a great idea because, private pharmacies are too expensive. Let's make generics, as if they were interchangeable, without really thinking through the implications of consequences. These pushes.
Yeah. I mean, look, you both of you are work in the cardiology area. I mean, have you seen a long-term longitudinal study of the mandatory, of mandatory genetic switching, when it comes to statins?
Zero.
Right? I mean, statin it's a standard of care right now, right? You turn 40, you put on a statin.
Correct. Now, the
Right?
statins are easy because you can have, in a way, you have immediate, I mean, there's a
Response,
bioassay in the
right.
person,
Yeah.
immediate response. But there's so many drugs that we use, we actually don't. It's so hard to distinguish, as you said.
Yeah.
From,
And I wonder, and I really wonder that I bet with no access to spreadsheets or data, I bet that the drugs where there is something to follow, an HIV viral load, a prograph level, an LDL. I bet in those particular cases, the companies are especially careful with that. But things like aspirin,
antibiotics,
antibiotics,
I mean, when we have AMR, I mean, how much of this is because of overprescription? How much of this is because of substandard drugs, right? So, there is a colleague of mine, Elizabeth Pisani, who works in the UK, and she's done some amazing work to show the impact of substandard drugs on antimicrobial resistance. It's just, it's a phenomenal area.
Really fascinating. Dinesh, tell us, we'll wrap up because we've taken up way too much of your time.
No,
but
right.
How is the story winding up for you in terms of, so you were,
So, I mean, there are two things that I try to do today. I mean, I think that a lot of what we're talking about today, right, is very relevant to us in the United States, but precious little has changed in India. So a lot of what, my focus is today, as a public activist, is to try and bring some systemic reform into the regulatory structure in India. Because, I mean, look, the country, that I come from has 1.5 billion people now, very close. And the regulatory structure is so poor, when I look at what the US FDA does, right? So the ways to go. And although, the US can certainly control and manage quality, those tools don't exist for the vast majority of people. I mean, my own family lives in India, right? So that's where a lot of my focus is. The other part of the focus is, again, trying to educate, lawmakers. A lot of times it's essentially sort of talking to them, in background and making sure they understand all the facets of, it's a fairly complex issue to try and explain. I mean, pricing is easy, right? Every election cycle, we talk about prescription drug pricing because it's an easy soundbite. This is not that easy to try and explain.
Yeah. Right. What a wonderful job you've done in explaining to us and being so patient with us this last hour and a half. So thank you so much, and thank you for having the courage and
thank you. That's very kind.
Gumption and it takes very special people to be able to have the insight to be able to see what's wrong, accumulate the data, synthesize it, and then go to the right people. You not only went to the WHO, then you went to Europe, then you went to the US, and so really, you're one of the heroes, true heroes of the story.
I was fortunate enough to have people like Raj in my life, right? Role models to kind of show you, what is the right way to do these things. I mean, sometimes you're naive enough to try and do things on your own, but if you're fortunate enough to have good role models and mentors to kind of guide you, it makes it a little easier to try.
Dinesh, can you tell us, we'll have it on the show notes, but can you tell the audience who's listening what your website and where they can follow you, follow your work?
Yeah, I mean, so normally I write, blog entries, but, they're geeky in the sense that you have to understand.
We have a lot of geeks in our audience.
So DineshThakur.com is the blog site.
Okay.
And, I don't write frequently, but when the issue comes up, I do write.
That's DINESHTHAKUR.com.
Yeah, and then on Twitter, I'm at D_S_THAKUR.
Wonderful.
I try and keep up with everything else that's going on.
The book that you're featured in, we should mention, is called Bottle of Lies. It's by Catherine Eben. Her Twitter handle, she's very active on Twitter.
Yes.
I see, and her Twitter handle is at Catherine KATHERINE Eban EBAN, all one word. Really great. Thanks again, Dinesh.
Thank you again. Yeah, wonderful talking to both of you. Thank you for having me.
Good night.
Right. Bye now.
Bye.